Hey team, Dr. Jake Gordy, and in my previous videos I discuss what is dementia and I describe different kinds of dementia, and then I undergo the discovery. I dive into the discovery of Alzheimer's disease by this fella here, Alois Alzheimer's. So now in this video we're going to continue that story and we're going to do some research into the pathophysiology of Alzheimer's disease, what is going on under the hood, but also some epidemiology as well. So let's jump into it. First of all, let's talk about the symptoms of Alzheimer's disease. So the symptoms are progressive, and so they progressively get worse and grow on top of each other. And the one thing I'll say is that they vary between patient and patient. The different kinds of symptoms, the order at which the symptoms appear are very variable between patient. So typically, I'm going to go through typically we see short-term memory loss, the ability to hold short-term memories go. Then we see older memories fade. Much more well-set memories will begin to fade over time as the disease progresses. We'll see mood changes, confusion and anger, and often we see this thing called night wandering, where in the evening the Alzheimer's person wants to leave and go somewhere, and that can be very dangerous for someone with Alzheimer's disease. We also see the loss of language. You could almost consider this a continuation of this memory problem. They start to forget words, but then they also forget how to put the words together. So they have actual really strong language loss. And then we can eventually see decline in motor function. Now, the decline in motor function is quite dangerous because we see a loss of ability to care for oneself, to get out of bed, or to eat effectively. And so after this, we often see death. Now, this on average, going through this process from diagnosis to death, on average is around 5 years. That does vary between country and diagnosis time. And some patients can live very long periods of time, but just on average, around 5 years. So short-term memory loss is a lot more like, I've forgot where I parked my car, where did I put my keys, that kind of thing. Older memories is quite interesting. So they seem to lose memory in chronological order often. So they revert back to older behaviors. So a really classic example is a patient whose older memories is putting the kettle on the stove, and newer memories is plugging in the electric kettle. As the disease progresses, they will sometimes, this is just an example, put a plug-in kettle on the stove because that's their memory of how kettles work. They've forgotten the new memories that kettles can work by being plugged in, and so they're reverting back to an older memory and an older behavior, which is putting kettles on the stove. Mood changes. Now, some of this might be directly due to neurodegeneration of regions that are involved in emotions, and some of this might also be due to symptoms. It's incredibly frustrating becoming forgetful and not being able to drive, and a loss of autonomy. So this is sort of all these symptoms are sort of integrated and shouldn't be considered in isolation. Language loss. Now, this is an example of a test we might give a patient. There's a few items in the list, but this is one of the items in the list that we give a patient who we suspect might have dementia or Alzheimer's disease dementia. So we will show the individual a wristwatch and ask him or her what it is, and then repeat for pencil, and then they get a point if they get it right. So patients eventually get this wrong, and they get the ability to name very common objects. They lose that ability. So it can result in very severe language loss. Motor deficits, and I mentioned frailty, imbalance, this kind of thing. But a real dangerous one is swallowing. They often can lose the ability to swallow effectively, and this can cause either food to come up and then go back down into the lungs, or food to go straight into the lungs whilst trying to eat. And that's very dangerous and cause pneumonia. And so the death is often very complicated. It can be directly through eventual neurodegeneration to the point that they get so frail that their body gives out. They've lost the motor function to survive. But often it's in combination with a different disease and a different pathology going on. So often it's pneumonia or bed sores or another thing as well that contributes to their death. And that's not to say that Alzheimer's disease isn't one of the major players in that form of death. So let's look at its global death, and I like to just do this to put it into context, right? we're just trying to evaluate what kind of burden are we seeing on the globe. So we have macro parasites, E. coli, influenza, malaria, staphylococcus, fungal infections, viral infections, COVID-19. Where does Alzheimer's disease sit in terms of global deaths per year? It's somewhere around here. It's quite a major player, somewhere around the 2.5 million mark right there. And that's just Alzheimer's disease. There are other dementias. And as we go through these videos, you'll consistently see actually non-communicable diseases, which are diseases that aren't infective, like heart disease and cancer and dementia and stroke. These contribute much more so to the global death toll than infectious diseases. Maybe this year was a bit different, but in general, when we look at down here, it's non-communicable diseases that are the leading causes of death in most countries, and Alzheimer's disease is no exception to that. Now, the error bars on this are very large. Some countries allow multiple primary causes of death on their death certificate, in which case often things like Alzheimer's disease and pneumonia can be listed. Some countries just have a single primary cause of death, and so the data can be a bit complicated to collect. In some countries, there are cultural differences around diagnosing dementia, and so the numbers about how many people in the world have dementia and how many people are dying from it is very hard to estimate, but we do know it's substantial. So what causes Alzheimer's disease? What is going on a molecular level under all of this? So here we have a brain. This is a cross-section of a healthy human brain. Then what I'm going to do is I'm going to put up a counter of neuronal death in a healthy human brain. So here we go. This is counting the rate of neuronal death. These could be my neurons right now. I mean, hopefully, because this is the rate that we would see in a healthy human brain. So we do see death of neurons. Now, the amount that are replaced is quite low, and that's sort of process called neurogenesis, or the generation of new neurons. That's very slow and very low throughout our lifetime. So largely when you lose a neuron, it's mostly gone forever. But we actually have more neurons and more computing capacity than we need, and that's called cognitive reserve. Our brain can figure out how to function perfectly fine whilst losing this number of neurons throughout time. And so we should really see no substantial decline in my cognitive function as I age, right? So that's just normal, healthy aging, is that neuronal death rate. Now let's look at the neuronal death rate in Alzheimer's disease. What we can see is considerably faster. It's around 4 times faster. And it's scary. And what we see because of that neuronal death is we see tissue atrophy. So we see the actual tissue of the brain shrivel away. Atrophy means to shrink, so we can see it shrink away here dramatically. Now, one area I really want you to look at down here, this is called the hippocampus. The hippocampus, which literally means sea monster, and it comes from sea horses. Their Latin name is hippocampus, and their tail has a swirl, and that tail looks like this swirl there, kind of, if you squint. So this is called the hippocampus, and this is where memory is encoded. It's stored temporarily, but long-term memory is stored out in the cortex, but short-term memory in the outer cortex. Short-term memories and memory encoding occurs through the hippocampus here. Now what we can see is that's completely gone. It's just a cavity in this Alzheimer's brain over here, and that's because that's where neuronal death typically starts, and then it spreads throughout the cortex, and we can see that with the symptoms, right? It starts here in the memory centers, but then it heads out to the language and the behavioral centers, and eventually the movement centers, causing movement dysfunction at the end there. And so the where the neurons are dying corresponds to where the symptoms occur and what kind of symptoms we see. So here, I'm just covering this briefly because we saw it in the previous video. Here are the histological hallmarks of Alzheimer's disease. It's this insoluble protein aggregate in the extracellular space. This slide was actually generated by Alois Alzheimer's over 100 years ago, and we found it in the basement of Munich University and took a photo of it, which is very cool. So this is one of the original amyloid plaques that were ever identified in history, which is super fascinating. And so this is an amyloid protein aggregate in the extracellular space, and it's quite large and it's insoluble. The other thing we see is a protein tangle inside of cells. So this is outside of cells, it's extracellular. This protein tangle is inside of cells. This is typically made up of a different protein called tau. So we see amyloid plaques in the extracellular space, and then we see tau tangles in the intracellular space. And the third histological hallmark is inflammation. So Eloise Alzheimer's described it as clusters of non-neuronal cells surrounding the plaque. So there's the plaque there, and there's a cluster of non-neuronal cells. This was drawn by Eloise Alzheimer's. And so this is neuroinflammation going on, and this is the third histological hallmark of Alzheimer's disease. Now I'm going to update that image. This is my image here. This is immunohistochemistry, fluorescence. And in green, we have a label for amyloid. In red, we have a label for microglia. These are the innate immune cells of the brain. They're a lot like a macrophage. And in yellow, we have where the microglia have swallowed, the red microglia have swallowed some green amyloid, and green plus red makes yellow. So in these yellow dots here, we can see really beautifully phagocytosed amyloid, which is fascinating and brilliant. And so this is the inflammatory response around those plaques. Now, what causes Alzheimer's disease? We don't know. We don't know. But we have a leading hypothesis. Now, the leading hypothesis is called the amyloid hypothesis. So a theory is something that is validated by all the data out there, and there's a large amount of data. So evolution is a theory because it's been validated by all the data. There's been no conflicting data out there, and so it encompasses all the available data. That's a theory. A hypothesis, there's some conflicting data, there's some counterpoints, there's some data that supports it, there's not a lot of data in general. That's a hypothesis. It hasn't yet reached the threshold of a theory. So the amyloid hypothesis states that amyloid comes first and initiates the disease. So let's jump into that idea. So amyloid species, so amyloid is a small protein, but it aggregates into oligomers, which means many subunits stuck together, and then it keeps aggregating into fibrils. I'm going to jump into this later. And these fibrils form insoluble plaques. So the amyloid hypothesis states that all of those amyloid species, not just the plaques, but everything, the oligomers and the monomers, something about all of these amyloid species triggers Alzheimer's disease. And it causes neuronal death and dysfunction. That's ultimately what we see in Alzheimer's disease. And it might do this in several ways. One way is directly. Perhaps the amyloid is directly toxic to the neurons. Perhaps it induces the neurofibrillary tangles on the inside made out of tau, neurofibrillary tangles made out of tau on the inside of neurons. And then those neurofibrillary tangles are what's causing neuronal death and dysfunction. And another one is that neuroinflammation. Maybe amyloid is inducing neuroinflammation, and neuroinflammation is toxic to the neurons down there. And if you want a bit more of a deep dive in that, I've got a video about how inflammation is damaging, and I touch on Alzheimer's disease in that as well. So where does the amyloid come from to initiate all these processes? Well, this is a synapse. Now, synapses are a connection between two neurons, and you can think of it as like, really, synapses are you. So the connection between neurons dictates their function and their activity, and it dictates your memories and your personalities and your movements, and everything like that is all controlled by the connections between neurons. A strengthening and weakening of those connections can be learning and forgetting and all that kind of stuff. So a synapse is the connection between two neurons. So here we have a synapse. Now, the synapse is generated through loads of proteins, but one protein thought to contribute to the formation of a synapse is the amyloid precursor protein, right? So this is a long protein that's in the membrane, and it has extracellular domains. So it's sort of stuck into the membrane, and it helps the synapse form, and it's called the amyloid precursor protein. Now, all proteins in your body undergo recycling. So they're chopped up, they're reused, they're rebuilt, right? Proteins don't have a long lifespan, so you need to chop them up, replace them, reuse them. You know, that's just part of what the body does. And so they might get chopped up. There's this membrane protein and a new one transcribed and brought out to the, and translated and transcribed and brought out to the synapse. And the old ones that have been chopped up are eliminated. Now, some of the elimination goes into the bloodstream to go around the body, get processed by the liver, for example. And other ones get eaten up by things like the innate immune cell microglia. So they eat up those chunks of amyloid. And this is what is going on in a healthy situation. Now, in an unhealthy situation in Alzheimer's disease, it proposes that there is perhaps an overproduction of these amyloid fragments, the amyloid precursor protein fragments. Perhaps there is an overproduction, and or there is reduced clearance. So perhaps in this example, we've got hardened blood vessels preventing the clearance of these amyloid fragments, and we have an inactive innate immune cell microglia, and it's not eating up those amyloid fragments. So either because of an overproduction and or an under clearance, a reduced clearance, something goes wrong in the production and recycling process, and it results in an accumulation of the protein fragments in the extracellular space. Now, one particular fragment is called beta amyloid. So the amyloid precursor protein is chopped up, and one of those fragments is called beta amyloid. Beta amyloid is very sticky, and it starts to stick together to other beta amyloids. Now, when you get a few of them together, we call those oligomers. And I'm going to jump into this a little bit later in a later video, but oligomers just means many amyloids stuck together. Oligo kind of means many or few. So an oligarchy means you are ruled by a few groups. That's the only other example I can think of. And so oligomers are a few amyloids all stuck together. Now, when you get lots of them stuck together, you get a fibril, and this fibril can become insoluble. So it falls out of solution, and we end up with these insoluble fibrils there. So the amyloid hypothesis states that these oligomers and fibrils can cause neurofibrillary tangles and cause inflammation. And now we're sort of starting to see the picture is a bit more complex, because inflammation can cause extra production and accumulation of amyloid. If you induce inflammation through infection or head injury, we can see an increase in amyloid pathology in animal models. Now, also, neuroinflammation seems to contribute to neurofibrillary tangles, and in fact, it doesn't seem like you need amyloid for this relationship. If you induce neuroinflammation, you'll get neurofibrillary tangles. So you can inject bacterial toxins or cause head injuries or infections or any number of things. If you get neuroinflammation, you'll end up with neurofibrillary tangles. Now, what's interesting is neurofibrillary tangles might also be inflammatory, so around and round they go, right? There's lots of feedback mechanisms there. Whenever you see these positive feedback loops, it is a sign of pathology, because we always like to get back to homeostasis. But if the inflammation causes tangles and tangles causes inflammation, you can see how that process gets a bit excessive and out of control. And it gets a little bit more complicated. So we've got amyloid inducing damage, we've got amyloid inducing inflammation, inflammation inducing amyloid pathology, inflammation also causing neurofibrillary tangles causing tissue damage. But tissue damage also causes inflammation. We understand that, right? If you roll your ankle, you get inflammation. So if neurons are dying, they're releasing damage-associated molecular patterns, they're releasing DAMPs, and that signals inflammation. So this is sort of a spiral as well, and amyloid might also induce neurofibrillary tangles. Now, most of Alzheimer's disease is sporadic. What that means is that there isn't a genetic basis to the disease. It's sporadic. It spontaneously occurs. We cannot predict at the age of 30 who is going to have Alzheimer's disease at the age of 80. Now, there are some factors that can affect your risk. Now, one such factor is genetic. So there are certain genes that increase your risk of Alzheimer's disease. What's interesting is many of these genes are associated with inflammation. Now, we also know head injuries and stroke can increase your risk of Alzheimer's disease and dementia, and that also is inflammatory. Obesity and diabetes increases your risk of Alzheimer's disease, and it is inflammatory. Aging itself is inflammatory. Your basal level of inflammation increases as you age, and aging is one of the biggest risk factors for Alzheimer's disease. And last but not least, infections increase your risk of dementia and Alzheimer's disease. And all of these have an inflammatory component. So I think this is where many of the risk factors enter into to the Alzheimer's disease system. I will say one other thing also enters this, I think, is vascular. So I mentioned there a little bit, if you have reduced blood flow or stiffened, thick blood vessels, your ability to clear amyloid is declined. And so many of these things are also associated with blood flow disruption. So I think that those two things are major contributing factors into how risk factors interact with your risk of Alzheimer's disease. Now, I'm going to tell you the evidence for the amyloid hypothesis, and I'm going to tell you the evidence against it. I think you're going to be completely convinced by the time I get through the evidence for the amyloid hypothesis, and then I think I might poke some holes in it and maybe convince you a bit more to be on the fence after I tell you the evidence against it. So let's jump into it. What's the evidence for it? Well, first of all, genetic. Now, I mentioned the vast majority of Alzheimer's disease is sporadic, meaning it's spontaneous and we can't predict it. But there's a very, very small fraction of Alzheimer's disease which are caused by single mutations, right? Now, so, and what that means is if I had that mutation, it would guarantee me having Alzheimer's disease, and it would give, and if I was heterozygous, it's a dominant gene, and so my children would have a 50% chance of having Alzheimer's disease. So, and there's a very few groups of families that have these risk genes, not risk genes, these genes that will guarantee you to get Alzheimer's disease. Now, here's the thing. We have only found families that are guaranteed to get Alzheimer's disease with mutations, and those mutations are in the amyloid precursor protein or the enzymes that cleave amyloid precursor protein. So that is very strong evidence that amyloid and the enzymes that digest amyloid have a key foundational causal role in Alzheimer's disease. Next one is Down syndrome. Now, this is a bit of an interesting one. So Down syndrome is when you have 3 copies of chromosome 21, and it comes with a range of symptoms and a range of biological processes going on. Now, it depends on which literature you read and who you cite, but certainly people would say that Down syndrome people have the majority will get Alzheimer's disease if they live long enough. Some people say 100% of them will get Alzheimer's disease if they live long enough and don't die of something else earlier. And they get that Alzheimer's disease quite young, not in their 70s or 80s, but more in their 50s. Now, what is interesting about that is on chromosome 21 is the amyloid precursor protein. So instead of having 2 copies of the amyloid precursor protein, you have 3, which causes more amyloid precursor protein being produced. And so you could end up with a buildup of amyloid in the extracellular space. And we see plaques building up in people with Down syndrome later in life. Now, fascinatingly, there are some forms of Down syndrome which are quite rare, where you don't get a full, complete third chromosome 21, and it's truncated. So a little bit of chromosome of the third chromosome 21 is missing. When a Down syndrome person has that third chromosome a little bit truncated and they're missing the amyloid precursor protein, so they've got 3 chromosomes, 21, but it's slightly smaller and it's missing the amyloid gene. So it's only got 2 copies of the amyloid precursor protein. Those Down syndrome individuals typically don't develop Alzheimer's disease, which is truly fascinating. And early mouse models of Down syndrome, we didn't see Alzheimer's-like pathology in those mice, but it turned out they had an incomplete replicate chromosome, and so they didn't have 3 copies of the amyloid precursor protein. When we popped the amyloid precursor protein gene back in, so they did have 3 copies, the mice then got Alzheimer's disease. And that leads nicely onto my third piece of evidence, which is animal models. If we take these genes that we know cause Alzheimer's disease in the human population, if we take those genes and we put them into mice, those mice become forgetful and they get amyloid plaques and they become worse at completing mazes, which we train them in, and so on and so forth. So we can measure the memory decline in these mice, and these mice do get memory deficits if we take this one gene here that causes Alzheimer's disease and we put it into a mouse. That's the evidence for. That seems really convincing, but let me hit you with the evidence against. First of all, there's a very poor correlation between amyloid burden and cognitive deficits. What do I mean by that? Well, what I mean is if you were to take 100 healthy individuals and 100 Alzheimer's patients, and you were to take their brains, section them, and quantify how much amyloid is in their brains, and then try to correlate the severity of Alzheimer's disease with amyloid plaques and amyloid burden, we would have a very poor correlation. Let me give you one example. There was a famous nun study in which a group of nuns kindly donated their brains to science. Now, one of the oldest nuns, she was over 100 years old, and she had excellent cognition, and she passed the cognitive tests fantastically. When they looked in her brain, it was full of amyloid. So her brain was full of amyloid, but her neuronal function was fine, and her cognitive function was fine, and she was very elderly. That's just an example of what I'm talking about. There's a poor correlation between amyloid and cognitive deficits. Next up, treatment failures. Numerous drugs have been developed that are capable of removing or reducing the amyloid from the brain. Many of them are biologics, they're antibodies that target amyloid, and we have successfully lowered the amyloid burden of a large number of Alzheimer's patients, and it did not affect the continued cognitive decline in these patients. One of these drugs was developed by Pfizer, who's in the news obviously for vaccines. And they spent $2.4 billion developing this drug only to have it fail in phase 3 clinical trial. I find that fascinating because people often accuse Pfizer of faking their vaccine data. But if they had the ability to fake clinical trials, I'm sure they would have faked their Alzheimer's $2.4 billion drug trial and made it work, right? If they could fake the data. The truth is, it's often all done by independent scientists who are blinded, and the company that does the blinding is independent, and then the people who do the stats on that data are independent. So Pfizer can't even get in there and interfere with the data, even if they wanted to. They are very rigorous and segregated from the process. And so this is why they have massive failures like this drug at $2.4 billion. Lastly, animal models. Now, I know I just said animal models was evidence for the amyloid hypothesis, but wait till you hear this. So if we put the amyloid genes and the enzyme genes into mice to induce Alzheimer's disease, we do get plaques, we do get neuroinflammation. We do not get neurofibrillary tangles, which is the second histological hallmark of Alzheimer's disease. We also do not get neuronal loss or substantial neuronal loss anyway. We don't get that atrophy. You remember that brain at the beginning of the video. Huge amount of atrophy goes on in Alzheimer's disease. We do not get that in the animal models. So what's going on? Yeah, we don't know. And this is why it's called the amyloid hypothesis. Now, I actually feel like the evidence supports the amyloid hypothesis, and let me explain just briefly why. So first of all, we now think we understand that amyloid takes decades to induce neuronal deficits and neuronal degeneration and cognitive deficits, right? And that actually explains a lot of what's going on. So the poor correlation could merely be, when did that person get the amyloid before they died? If we look at our 100-year-old nun who had fantastic cognition but lots of amyloid, she might have got that amyloid buildup in the last 10 years. But if it takes 20 to 30 to 40 years to get symptoms following amyloid buildup, that's exactly what you would expect, really poor correlation. It's like if you took a bunch of 85-year-olds and told them to start smoking. We probably would see that there isn't a correlation between the smoking and lung cancer risk because they're 85 years old. You need to smoke for decades to build up your risk of lung cancer. And so if you were to suddenly try and draw a correlation between the number of cigarettes you had yesterday and lung cancer, it wouldn't work for people who took up smoking yesterday, right? And this is the same with amyloid. If you got your amyloid recently, over the last decade or so, we wouldn't actually expect that to cause neuronal loss and neuronal deficits. Now, this time it takes for amyloid to cause this process also explains the treatment failures. We took people with Alzheimer's disease, we removed their amyloid and looked for cognitive improvement over a year or 2 years. You wouldn't actually expect to see the benefits of that unless you took the amyloid out when they were 50 years old before they developed those cognitive symptoms. It might take decades to induce the problem, and it might take decades to get rid of the problem. So anti-amyloid treatments might only be effective in the very young, which is why, and by that I mean middle age, 50 years old, 40 years old, which is why we're desperately working on early diagnostic techniques for Alzheimer's disease. And we're seeing some good results, particularly in cerebral spinal fluid measurements. Lastly, animal models. This again comes back to time. Mice typically only live to 1.5 years. However, most Alzheimer's studies are often done in a year. So the mice only have amyloid in their brains for maybe 6 to 8 months, right? And that might not be enough time to build up the neurofibrillary tangles and the neuronal loss. And evidence for that is we've now started to create transgenic rat models that replicate the Alzheimer's disease symptoms. Now, rats live longer than mice, and what we're seeing is neurofibrillary tangles and neuronal loss in the rat models of Alzheimer's disease. So it could be to do with time, where you need a lot of time to develop that process of amyloid to inflammation, to neurofibrillary tangles, to neuronal loss. You need time for that process to happen. Brilliant. So that was an introduction into Alzheimer's disease. Up next, I'm going to go into how does silver staining work. Now, silver staining is a histological technique as well as a protein technique that Alois Alzheimer's used to recognize those histological features of Alzheimer's disease, but it's still used today because it's such a good technique. So I'm going to jump into silver staining next.